Hi everyone, welcome to our October's edition for the Recon Community Meeting. Today we are joined by Enrico Calore, who has had quite an unconventional route to the field of synthetic biology, engineering biology, and biomedical engineering in general. Because he first started his master's degree in aerospace engineering, and he worked a few years in the field there in automotive engineering, as far as I understand it. But then he transitioned into doing a master's in science degree in biomedical engineering, synthetic and systems biology at Duke University in North Carolina, USA. He's particularly interested in how we can apply synthetic biology to develop practical and translational solutions in medicine and biotechnology, make it more accessible around the world, and to help promote global health. And I believe he plans to continue in this direction for his doctoral studies moving forward. But more recently, he has been working on a pipeline for producing thermostable molecular biology enzymes in-house. So very much in line with what we do at Recon here. And the plan is to make it faster, cheaper, and easier for labs to access these core reagents. So he'll be presenting about the work he did for this today. Published earlier this year, I believe. Um, and in this area, he's covered key reactions like DNA amplification, transcription, and assembly at just a fraction of the current commercial costs. But with that, I'll just pass the mic over to Enrique to tell us a bit more. Thank you and welcome to the community, Enrico.
Hi, everyone. Thank you so much. I'm very happy actually to be here and to connect with you all. And yes, today I'll be presenting our recent work titled Instant Enzyme, System Engineer Approach for Rapid Low-Cost Production of Thermostable Molecular Biology Enzyme. And this work was recently published in BioResource Technology and was led by Lynch Lab at Duke University, USA, in collaboration with different people, including myself. And by the end of this presentation today, we'll see how to build a unified workflow that simplifies enzyme manufacturing, make it easier, cheaper, and more accessible, particularly for those labs with middle low income. So to frame the motivation behind this work, in synthetic biology, everything revolves around this design, build, test, learn cycle. And the build phase, which allows for the physical assembly of genetic system, heavily depends on enzymatic tools. And among these, thermostable enzymes stand out as a crucial component for DNA amplification and assembly, and really helping us to turn concept into something that we can actually test. Despite being essential to nearly every molecular biology workflow, thermostable enzymes remain costly and difficult to access. And here is a plot that is showing basically the catalog prices from Thermo Fisher Scientific for this October 2025. And on the x-axis, we have common enzymes such as DNA polymerases, reverse transcriptases, and ligases. And on the y-axis, the heroes per reaction. And you can spot that even basic even basic reagent like Taq polymerase costs around 70 cents per unit, which adds up very quickly. For example, in our lab with 5 to 6 people working on the bench, we were spending something like 10 to 20K per year just on polymerases, which is a lot. And in-house production of this enzyme is possible, but it's rarely practical because usually yields are low, workflow are complex, and reagents are expensive. So as a result, most labs remain dependent on these commercial suppliers, and these high costs continue to act as a barrier to innovation. So there is a clear need for a simpler, scalable, and cheaper enzyme production method. So we are looking for a standardized framework which is capable of reducing enzyme prices by at least 1 or 2 orders of magnitude compared to the current costs. So how can we do that? So what you can see here is a typical workflow for recombinant protein production. We have fermentation, cell lysis, and chromatography purification. And with this pipeline, usually it requires an expert operator, 8 hours of hand-on work, specialized equipment with expensive reagent. And this results in a process that is low resource intensive and hard to scale. And because optimization usually target individual step in isolation, improvement tend to be incremental with limited impact on the overall efficiency of the system. So instead of optimizing each step in isolation, this work reimagine the entire process as a single integrated system. So the goal here is to link fermentation, lysis, and purification into a single streamlined workflow. So in this setup, we use autoinduction to trigger protein expression directly during fermentation. We have engineered cells to self-destruct just at the right time. And we use precipitation to recover the enzyme completely bypassing chromatography. And with this system in place, the goal is to make enzyme production faster, simpler, and more accessible to any lab, regardless of scale and resources. So now we're going to explore each step of this pipeline, starting with the autoinducible fermentation. So in traditional fermentation, protein production is manually induced while cells are still dividing. And this means that growth and expression happens at the same time, forcing the cell to split their energy in between replication and protein synthesis. And this creates metabolic stress that lower biomass and yields. So in contrast, we are working with two-stage fermentation. And here, growth and expression are completely independent stages. So first, cells grows to high density into under nutrient-rich conditions. And then when phosphate is depleted in the media, autoinduction naturally triggers protein expression with no manual input required. So this two-stage design remove timing error, metabolic stress, reduce metabolic stress, and allow for higher biomass and yield, and enable reproducible expression across scales. So to see how it works in practice, here you can see protein gel, where we are comparing Pac polymerase expression with traditional and two-stage fermentation. And you can see we have a high level of expression with pretty high, with 1 gram per liter, which if we, in practical terms, that means that with that expression in lab, we can use a 20 milliliter flask and this cover for about 4,000 PCR reaction. And this is equivalent to 2,500 worth of commercial tag with the prices we saw before, which is a huge saving. And this is achieved without any fermentation optimization. And so let's move to the next step and discuss how cell autolysis work. So basically, the autolysis system is built directly into cells themselves, meaning they co-express all the components needed for self-destruction. So before induction, nothing happens. So the cell just grows normally. So after induction, when phosphate is depleted, 2 key enzymes are produced, the lysozyme, which degrade the cell wall, and the benzonase, the nuclease, which degrade the nucleic acids. And as you can notice here, those enzymes are within particular compartment of the cell. The lysozyme is expressed in the cytoplasm and the benzonase within the periplasm. And because of their location, they are inactive during cell expression. And after expression, basically the cell is ready to autolyze until a trigger is induced. And this trigger is the addition of a detergent and a freeze-thaw cycle. And this basically release the enzymes. So the lysozyme degrade the peptoglycan wall, and the benzonase degrade the DNA and RNA. And the result is a clean lysate, which is ready for purification. So what's elegant about this design is that the autolysis system is seamlessly built into the same autoinduction framework that drives enzyme expression. So as we discussed before, during cell growth, nothing happens in terms of expression of enzymes and lysozyme and nuclease. But after the phosphate is depleted, the system switches to the production phase, co-expressing the target enzyme and the lysozyme and nuclease under all the same control. So to test how this works, how well this works, so we compare 2 strains, a control and an engineered strain, expressing lysozyme and nuclease. And both were induced to express GFP as a reporter for protein release. And what you can see from this plot, when treated with detergent, the autolysis strain release increased amount of GFP over time, reaching a plateau at around 1 hour. And the next step was to test the system at different temperatures after adding the detergent. And as you can see, the highest release was observed at -20, -60 degrees, where the autolysis strain released almost all of its GFP content into the miniole, while the control strain released virtually none. So this shows that the engineered cell can self-destruct efficiently without the need, for example, of mechanical lysis or any additional chemicals. And the next step was to confirm that the expressed nuclease was active and sufficient to remove the host DNA. And to do that, we compared 3 strains, one expressing active nuclease, one expressing inactive mutant, and another one with a knockout strain with basically the nuclease deleted entirely. And as you can see on the gel, the complete DNA degradation occurs only on the strain expressing the active enzyme. And this confirmed that the autohydrolysis module is fully functional and capable of degrading nucleic acid directly within the cell. So now we focus on the precipitation step. And basically here we apply the whole workflow for the purification of a ACT polymerase. And to recap, after expression, so we basically harvest the cell. We resuspend with a buffer containing a detergent. Then we freeze for an hour, and then we thaw the cell for 2 hours to achieve full autolysis. But when we applied that to TAC, we noticed that we had DNA traces in our lysate. And we believe that this is due because during the thaw cycle here, traces of DNA may be amplified by TAC because TAC is still active at that temperature. And so to fix that, we basically add another step, just 15 minutes at 60 degrees that fixed the issues. And although this mechanism wasn't fully characterized, this simple heat step was sufficient to provide a DNA-free preparation, improving the robustness of the workflow. So moving forward, we had another issue. So basically, we found that the purified enzyme still retained traces of nuclease activity. And we confirmed that with this experiment. We basically incubate plasmid with our instant TAC and control a neb TAC. And you can see everything is fine. You can see the plasmid band at T0 after basically after the sample was made. But after 16 hours, you can see that the plasmid that was incubated with our TAC polymerase fully degraded compared to the control. And basically, this confirmed that there was residual nuclease activity in our sample. So to ensure that the enzyme is suitable for downstream application, we needed to figure out a way to achieve nuclease inactivation. So to do that, we focus on benzonase, which is the nuclease we are expressing in our cell. And we saw that with our workflow, the heat that we apply is not sufficient to degrade the nuclease. We know that benzonase relies on disulfide bonds for structural stability and catalytic activity. We also know that adding a reducing agent can break these bonds, but this effect is reversible. So our hypothesis was to combine heat and reducing agent together, hoping to push the enzyme into an irreversible state and precipitate out of solution. And this concept basically laid the foundation to our design of experiment that we will be discussing now. So basically, to systematically identify the optimal precipitation condition for inactivating and removing residual nuclease, we used the DOE experiment. And so basically, we tested the 38 condition, varying not only heat and reducing agent. We also added ammonium sulfate concentration, pH, and time. And basically, we built this multifactorial experiment, and which we evaluated for residual nuclease activity. And our goal was to identify those conditions that basically inactivate nuclease while keeping TAC polymerase soluble in solution. And these conditions are those one highlighted in green. And so basically we use this condition to move forward and finally purify the TAC polymerase. And this enzyme was validated through key analysis. First, we use protein gel to assess the workflow, the purity across the workflow stages. And you can see there is a progressive enrichment of the TAC band with the removal of OX contaminants. And second, we compared enzymatic activity of against a commercial TAC control. And even here, you can see that we achieve identical amplification across enzyme dilutions. And third, we use a qPCR assay to analyze the melting profile. And even here, you can see that the peak aligns around 85 degrees, confirming that the enzyme amplifying the same target with the same specificity. So together, these results confirm that our simplified workflow function is deliver a functional equivalent enzyme to the commercial standard. So after with the DOE data in hand, we decide to model and understand how each parameter influence nuclease inactivation. And so here, basically, the nuclease activity was modeled using a second-order polynomial fit to the DOE data. And the results were visualized as a contour plots across the experimental factor space. And to better understand this plot, you see we have the temperature on the top, and we have ammonium sulfate as a variable in this case on the y-axis, the H range on the x, and the constant are here on the right, the reducing agent and the incubation time. And then the nuclease activity is followed this legend where the lower nuclease activity is with a cooler color. And this specific plot, for example, shows that at higher temperature combined with moderate soil condition consistently minimize the residual activity. And expanding this analysis across pH, reducing agent, and incubation time, we were able to build a multi-dimensional design space for complete nuclease removal. And this design space was helpful, and we use it as a reference for purification of other enzymes. And you can see these, the black dots represent the final purification condition for the enzyme in this work that we will be discussing later. So after optimizing the precipitation condition, the next step was to test whether this system could be scaled up and automated within a bioreactor. And in this setup, we will have the purification, the cell lysis, and the precipitation, and the expression of the enzyme, the lysis, and the purification happens all in the same vessel. So you can see here in the plot that it's showing total protein and TAC concentration, that how they evolve over time. And here is the temperature profile that basically guided the entire workflow. At 60 degrees, the cell lysis and nucleic acid degradation begin. At 37, enzymatic digestion continues. And at 70, heat precipitation remove host contaminant, and TAC remains soluble in solution. So on the right, you can see the protein gel that confirms this progression and showing enrichment again of the TAC polymerase, and through each temperature stage from the purification from the expression to final purification. And together, this result demonstrate that we can streamline a bioreactor workflow, achieving high purity and yield with potentially no manual intervention. And now that we have demonstrated this workflow is working for TAC polymerase, now we ask ourselves if we can apply this workflow for other enzymes, particularly ligase, reverse transcriptase, and high-fidelity polymerase. And we chose these enzymes because they represent the core biochemical reaction of assembly, transcription, and amplification. And basically, they provide an ideal benchmark for assessing the general applicability of this pipeline. And also, they eventually provide a tool that equip any lab to make these enzymes in-house at a fraction of a cost and turnaround time. So let's start discussing the ligases first. So for the ligase purification, for all the enzyme in reality, we are going to apply the same workflow and the same optimized DOE condition for the precipitation of nuclease, and see if we are successful in purifying those enzymes. And if you remember, when we saw the optimized DOE condition, we found more than one. So what we are doing here is just testing those conditions with the ligase. And you can see here, for example, that the addition of reducing agent is beneficial for the ligase because we achieve higher purity and higher yield. However, when we increase salt temperature and salt, that was beneficial for TAC. Here is detrimental because you can see we have a drop in the purity and the yield, ending with losing the enzyme. So these results confirm that some precipitation principle identified for TAC can be adopted for other enzymes, but there is a crucial need for empirical fine-tuning when we transfer those optimized conditions across the enzymes. So after purification, we validated instant ACK ligase, both structurally and functionally. And on the left, you can see this protein gel showing again strong soluble expression and clean purification. And on the right, we use S.LSA to evaluate the ligase ability to assemble DNA fragments. So basically, we use our ligase and commercial control, and we basically construct our plasmid with antibiotic marker. We transform in E. coli and grow in the colonies on plates, knowing that each colony is represent a successful assembly event. And you can see here that after 15 ligation cycles, we were able to demonstrate that our ligase is working, even though we are achieving 1/3 of the colony count when compared to the NAB ligase. However, this is still demonstrating that our simplified process preserves functional activity of our instant ligase. So next, we try to apply our workflow to the reverse transcriptase. And here again, we apply the same conditions, DOE optimized condition for nuclease precipitation. And we noticed that basically we were losing the enzyme. Particularly when in a reducing condition using reducing agent and rising the temperature, our enzyme basically were precipitating out of solution. But because among the DOE condition identified, there was also a condition without the addition of the BME, the reducing agent, we just applied that condition and we solved the issues and we achieve purity and yield at a high level of purity and yield, particularly 99% purity at 80 degrees. And this result confirm again that individual enzymes respond differently to specific parameters, in this case, reducing conditions. And this highlight, of course, the importance of tuning purification chemistry to each enzyme structural stability. And again, we validate our enzyme. So here in the protein gene, you can see we were able to recover high purity reverse transcriptase. And then functionally, the enzyme perform on par with commercial BOCA scientific standard. You can see here, this is a qPCR assay. The CQ values scale linearly with the enzyme concentration and overlap precisely with the commercial control. And this confirm equivalent reverse transcriptase, transcription efficiency, and quantitative accuracy. And finally, if we focus on the high fidelity DNA polymerase, and here, again, with the same workflow and optimized DOE condition for the precipitation of nuclease, we detect residual nuclease in our samples. So basically, the DOE condition failed for this enzyme, and we believe this is due because this enzyme was expressed in much lower level compared to the other enzymes, and probably this impact on the precipitation selectivity and performance. So to fix this issue, basically we extended our DOE condition. We broadened our temperature range, pH range, and salt range, as you can see here. And we basically built 29 new conditions and hoping to find at least one condition that we actually found that remove nuclease activity. And these conditions result in 50% purity and 60% yield, which is actually a suboptimal result, let's say. But we move forward, and after purification, you can see that we were able to still achieve a selective enrichment of fusion polymerase, confirming recovery without chromatography. And then we test the PCR amplification against a commercial control, an AB commercial control. And then you can see that the enzyme perform identically to the commercial NAB enzyme across the same activity range. So we confirm again that this workflow consistently deliver nuclease-free, fully functional enzyme, demonstrating the robustness and adaptability of this pipeline. So to summarize, we developed a unified workflow that enable in-house enzyme manufacturing in a fraction of the time and cost. We use this workflow and scaled up in a temperature programmed bioreactor. We were able to demonstrate that expression, lysis, and purification can happen in the same vessel with minimal human intervention, confirming its potential for larger scale production. And we used the design experiment framework, and we systematically tuned temperature, pH, salt, and reducing condition to achieve complete and consistent nuclease removal from our lysates. And finally, we extended this standardized pipeline across key thermostable enzymes, everyone showing commercial grade performance at a fraction of the cost and labor. And of course, this workflow comes with important constraints. Because it relies on thermostability, it is only compatible with enzymes that can tolerate elevated temperature. For low expression, like we saw in fusion polymerase, we observe lower purity and yield. And some residual host protein and salt can remain in the lysates. So they can interfere with the sensitive downstream application. And long-term stability of the enzyme has not been systematically evaluated. And since individual enzymes respond differently to specific parameter, as we saw before, the purification chemistry need to be tailored to each enzyme structural stability. And finally, the bioreactor purification works and allows to save a lot of time potentially, but scaling to larger volume can be challenging due to heating cost and temperature uniformity. And that's the end of the presentation. I just want to thank you, Duke University and Lynch Lab, for the opportunity of working on this project, the funding that we had, the support of our effort, and for you all to be here listening to my presentation. So thank you very much. And I'll be happy to answer any question, if any. So thank you.
Thank you so much, Enrico. Looks like folks in the chat, well, folks online are very excited about this work, and with every right to be. You already have a question in chat. Maria, would you like to voice up to read it, or do you want me to read it for you?
You can feel free to read it if you want.
Sure. I'll pass it on to Maria because she has her hand up.
Hi, how are
If
you?
not, okay.
Yes. This is Maria. Can you
Hi.
hear me?
Yes.
Yes.
Yes. Hello, Maria.
Thank you very much for your presentation. I wonder what software do you use for the design of your experiments?
You ask the support for the design of the experiment?
Yes.
You mean what tool we used, or what kind of support you're referring to?
The software that you use for
the software. The
yes.
software is GMP.
Okay, thank you.
Of course.
I'll go to the chat from Kajalcha. Sorry if I'm pronouncing your name incorrectly. Sorry, you said thank you so much for the clear chat. I think we had Ali in the chat next, and then we'll go to Pablo afterwards. So Ali, would you like to ask your question?
Sure. I'm coming through, right? I'm curious if the strains are available for others to use as well, these auto-inducing, self-destructing ones, and what you think about people modifying the system for other enzymes with other plasmids. How easy do you think that transferring from a column-based system to this would be? Thank you.
Okay. Hi. Hello, Ali. So basically, the strains are actually available for purchase because the guys that the first author of this paper actually found a company which is called Rogue Biotechnology. I can write it in the chat. And basically, you'll find that strain is found a company based on this technology. And the second question you asked, basically, if I understood correctly, is how easy it is to engineer this strain. Did I get it correctly, or you were asking another point?
Yeah, basically that's and how easy you think it would be to adapt column-based enzymes into the system with the phosphate induction, etc.
Okay. I have actually some slide that I can support this. So basically you have to act on 3 levels. So basically you have to engineer strains, to engineer the media, and basically to engineer plasmid, make your plasmid sensitive to autoinduction. And so basically here, so what the lab did, they start from a well-characterized K12 E. coli strain, and basically they for the strain engineering. This is at the big. I'm treating just one level first, the strain engineering. So basically they start from this strain, and they introduce genetic deletion to increase yield and stabilize autoinduction. Basically reducing overflow metabolism and energy use. And as you can see here the 2 strain the lab is using, these are the deletion that they are made. And this is the first step. Once you engineer the strain, you then move to engineer the media. So basically this strain works well in minimal media, and minimal media is good because it's cheap, but it doesn't work very well when interfacing with other media that a lab may use, which usually is nutrient-rich media like LB. Because when you inoculate in minimal media, the cell experience a sort of lag, growing lag, which is not beneficial in a workflow because you may lose time eventually. And so the lab, this is one reason to engineer the media, and another reason is that you have to provide a media with limited and self-depleting phosphate, because you have the phosphate need to be depleted at a certain time to ensure that the workflow is reproducible, is standardized, and really happens, And so the lab basically engineer a media, again, with a DOE experiment. You can see here all these factors are basically the main component of the medium they tested. And basically the phosphate is provided by these 2. And similarly to what we discussed before, each column is an experiment with a combination of this factor. And they tested how well this media will enable a strain, this one, which it is one of the strain we introduced before, how well they express the GFP, and how well the biomass, the level of biomass. So basically, they were measuring the GFP expression and DOD, and based on the result, they select the best combination, and they named this media autoinduction growth, this AB media, and that's the media the lab is using basically. So you have the strain, you have the media, and the next step is to engineer the promoter. So basically the plasmid with the correct promoter. And to do that, the lab basically investigate promoters that were available in literature. And this promoter basically leverage a native pathway in E. coli where basically E. coli is able to sense the presence of phosphate in the environment. This POR-B system, where POR is a membrane receptor and POB is a transcription factor that is activate when the E. coli sense low phosphate. And so basically the lab searched for POB regulated promoters, and they selected based on the simplest version. So the one that has minimal regulation, particularly those that rely just on sigma 70 sigma S factors that are easier to engineer. And they tested with via IAB again, the immediately discussed before. And you can see here in these graphs, basically you can see all the promoter they tested on the x-axis. And They tested both in minimal media and the auto-action broth, the media they engineered before. And basically, they select the best promoter based on the GFP expression, minimal leakage. So the leakage is the black bar here, and the highest OD values. And based on this, they select a promoter that is called EBD, this ones. And they did another engineering step because they check. So this is the EBD promoter sequence. They check this native sequence and also a truncated version, where the truncated version, basically they cut off this part, which is one of the POB binding box, which is also repressed by this NSR, this repressor. They basically cut off this and check again the performance of these two promoters and find out that this one performed best, the shorter version. And they check again against the GFP expression and OD, as you can see here in this graph. And that's the promoter the lab is using. So to recap, if you want to run the two-stage auto-induction, you basically need an engineer strain, an engineered media, a media that is suitable at least for phosphate depletion. And one of these POB regulated promoter, and EBD is the one that the lab is using that based on this experiment, basically. This version is optimized, let's say. And I don't know if I answered your question.
Yeah, it was perfect. Thank you so much.
Of course. Happy. It was helpful.
Cool. So I'll pass on to Pablo, invite you to ask your question, and after Pablo, it'll be Ben.
Okay, hello, thanks for your work. Really, really interesting. Fairly simple question. How is the system tuned in order to get high levels of your protein of interest before start with the induction of the lysis proteins and before getting the bacterial lysis? That's the first question.
Yeah, so basically, when you use, in my experience in the lab, I try to express different protein and with this system, and you can notice that you have some variability in the level of expression. Some protein express very well, like GFP. Other protein may express less well. So basically, when I was doing my experiment, I don't really use, there was not a qualitative check after expression, unless, it really depends on my on my goal, what I was what I wanted to achieve. So, but in reality, as a protocol, there is not a quality check after the expression before the cell autolyses, because the system is robust and usually always work. Even though, as I was telling you before, you may experience different level of expression, depends on the protein that you're working with. But the system is very robust, and in my experience, it never failed, basically.
Okay, and the second and final question is, which scale did you use for the optimization part before scaling up? I mean, the DOE experiments and so on.
So what you're asking specifically for the DOE? What you're asking?
Which scale, which volume of culture or in which scale did you work?
Yes. So for this, we were working on flasks, but flask with 20 milliliters.
Okay.
But it should be also be tested on microplates and and also 1 liter bioreactor. So it should be scalable. And but if I think for this particular experiment, I believe they used the flasks.
Okay. Thanks.
Of course.
Thank you very much.
Cool. Ben, I think you had a couple of questions in chat. Would you like to voice them to Enrico? And after Ben, we'll go to Vincent.
Sure. Can you hear me okay?
Yeah.
Okay. I was just confirming what component in your minimal media is actually providing the phosphate. I couldn't see It wasn't obvious to me on that slide.
Yes, in the minimal media, I think they use they provide. I'm not sure about I'm not sure which component they added, because I'm not used to work with that, so I never prepared actually in the lab. It was used just for this kind of experiment that I didn't perform. So I don't want to provide you incorrect information. So I'm not sure which component they use for the phosphate in the minimal media. Sorry about that. Sorry.
Okay, which paper would that be described in?
Yes. You should. So it is called. I'm gonna look for it. If you type two-stage autoinduction in Google, you will find it straight away.
Great, thank you.
Of course.
Over to you, Vincent, for your question, and then we'll jump to Ganesh in chat, but Vincent first.
Okay, thanks. Microphone check. Can you hear me?
Yes.
Good.
Okay, great. So Enrico, thank you so much for presenting. That's very cool work. My experience with producing recombinant polymerases was just boiling a liter of cells for PFU about 20 years ago. So I really appreciate you doing this in a much more systematic method. I totally love the work you did with the polymerases. What I'm interested in is how you're going to take this forward to work with non-thermostable proteins, right? How you're going to actually try to clean up these backgrounds when you have these other issues. I just wanted to point out there was a paper from about 20 years ago about a single protein production strain that was produced in E. coli using toxin antitoxins that I'd like to share with you, that I think in the future, if you move forward with this and start looking at non-thermostable proteins, could actually be of benefit, because it makes it to where all other proteins basically stop being produced as you're producing your own, which may actually help clean up some background. But essentially, I just wanted to say thank you.
Oh, thank you so much. I appreciate that. Yes.
No problem at all. Thanks.
Always good to be appreciative of the work that researchers do, huh? So I'll move to Ganesh. Would you like to voice your question? And no worries if not, we can also voice it for you. So Ganesh posted in chat, were the nuclease and lysozyme enzyme genes integrated into the genome, and were the inducers the same?
You're talking about the autolysis, how the autolysis works in the gene?
I believe so, yes.
Yeah, I have also a slide for that. I think I can share. Yes, so basically, okay, I think this is the right slide. Yeah, so basically this is, can you see the screen? Because I think I temporarily unshared before. Can you see the slide?
We can see this screen. It's on slide 39.
Oh, that's perfect. Yes. So basically, this is the figure that we discussed before in the presentation that we are a little bit familiar with. So basically, after expression, the cell co-express these enzymes in the lysozyme in the cytoplasm, and the benzoname is the periplasm. So I think the question was how we can direct, I guess, what is the gene cassette, the gene cassette, and how we can direct the benzonase on the periplasm, I guess. So basically, this is the cassette we used. So you can see here there is the promoter we discussed before, phosphate inducible. The promoter that triggered after phosphate depletion, and these are the genes. So basically, we have the lysozyme gene and the nuclease gene, and particularly to direct the nuclease to the periplasm, you basically add a signal peptide on the N terminus of the nuclease, and basically this is enough for the to trigger the SEC pathway. So basically, when the nuclease basically literally exits the ribosome, it is bound by a protein that prevent it to fold within the cytoplasm, and it is directed outside on the periplasm where it can fold. So this is I'm not gonna I don't think it's beyond the presentation to go over the detail, but this is the SAC basically pathway. As I was anticipating before, when the protein just literally exits the ribosome, there is this SAC-B protein that binds and prevent the nuclease to fold. And then there is a SAC-A protein that is also recruited, which is an ATPase that promote and help the protein to get out of the inner membrane. And then at this stage, this signal peptide is cut and the protein fold and the disulfide bond forms. So to answer the question, basically you had to insert the cassette we use this cassette in the we actually insert this cassette in the genome and in the OMT locus, and this locus is natively is occupied by this perismatic protease that we deleted because it can also delete the recombinant protein in the periplasm. So use basically recombinating to insert this cassette in the OMT locus of the genome of the E. coli, and that's basically how it works. I don't know if I answered the question, but that's at the high level how it works.
Yeah, Ganesh says, cool, he got it. So hopefully that did answer your question. I did see Felucio had a hand raised earlier. I don't know if you wanted to come back to you, but I'm going to jump to Mahe in chat. Would you like to voice your question, Mahe?
Do you want me, Felucio, do you want me to go ahead?
Sure, yeah, Felucio, do you want to go first, and then we'll go to Mahe, and then Adrian after.
Don't worry.
Yeah, I just had a quick question about the purification system. I don't know whether you mentioned when it was presented, or maybe I missed something. I was just wondering whether you guys try to do some dialysis, just to get rid of the contaminant, because you mentioned that some of these contaminants can actually affect sensitive proteins. So I was wondering if there's a cheap dialysis bag that you guys can, you guys just try to reduce the contaminant, basically. I know you did most of these things just to lower the cost of your final product, and I don't know if there are some if there are things that you if there are other things that you guys try just to reduce the background chemicals.
Yeah, so basically we didn't because when we were using this workflow targeting a particular enzyme, and it basically, with some challenges that we discussed before, we always achieve we were able to produce a functional enzyme every time, the enzyme that we were targeting. And so we never use any dialysis after purification, because we basically demonstrate that the enzyme was working was functional, and we didn't implement this enzyme for any downstream process. So basically, to answer your question, we didn't introduce that step, even because one of the purpose of this workflow was to be streamlined and eventually automatized, and maybe dialysis is not, to my eyes, not might be fitting this perspective, but yes. So I don't know if I answer your question, but I believe so that was the reasoning behind the workflow.
Okay, thank you.
Of course.
Great. So I'll go Mahe next, and then Adrian.
Okay, thank you so much, Yankei. And thank you a lot, Enrico. It was a super nice presentation, super nice work.
Oh, thank you.
So congrats about that. And it's a super small question. Did you say at how much did you scale up? 20 ml, I said?
Oh, okay. So we scale up to up to 1 liter in the bioreactor.
Okay.
Yes.
Okay, perfect. Thank you so much.
Of course.
Adrian, over to you.
Yeah, thank you, Enrico. That was a great presentation, and also I like the fact that you managed to integrate all these steps into a fairly fairly simple process there. I have two questions. On the slide in here, you mentioned something that you inserted something into the genome. So I was under the impression that it's only the plasmid that's necessary for doing this. And the second question I have, I looked at that homepage in there and I see 3 plasmids. There's E. coli DIY instant TAC. There's 6AL for proteins with disulfide. So how many plasmids would you need for, is it like a multiple plasmid system or is just a 1 plasmid system for, and I don't see, I see just the TAC in there. I don't see the fusion, neither the ligase nor the reverse polymerase.
Okay, so for the autoinduction, you just need 1 plasmid with the EBD promoter and the gene that you want to express. And so if you, I don't know, because because the company is pretty new, they are the website is renewing very fast. I saw it is, I don't know, I saw maybe it's, I don't have in mind actually the layout and the product that they are selling at the moment. But what was the other question regarding the plasmid? There was some plasmid that were selling. For sure that maybe they are selling the TAC polymerase. That is the one, is basically done with the same principle we discussed before in this presentation. But I don't know if actually the current products layout of the company. So do you have a specific question on that regard?
Yeah, actually, I posted the URL for the 3 strains that they have in there. And my first part of the question, which was kind of separate, was because you were mentioning something inserting into the DNA, and I was wondering if it's just the plasmid DNA or you're talking about the genomic DNA.
Well, this is genomic DNA. So basically, this cassette is inserting the genome, but so when you use the strain that the basically autolysis strain is that's already done, but you need this promoter to on the plasmid that you are transforming in the cell in order to express the gene, because this is actually the promoter that is sensitive to phosphate depletion.
Okay. Thank you very much. Great work, by the way.
Of course. Thank you so much.
I think we are coming to time on the chat. I thought I saw another question from Ganesh saying to point, if Enrico, if you could point folks to other related work. And I think folks have shared various things like the autoinduction media and the Roche bio links in chat. But also feel free to use the forum link directed to add any more questions, any more feedback, any more resources. I think this is definitely of a lot of interest to everyone here. So thank you so much, Enrico, for sharing with us today, and for everyone else for your insightful kind of comments and queries. One last question I can see in chat is from Ben. Can you show the slide on the recombinering again, if that's okay, Enrico?
On the recombinering,
yeah.
So is this one?
So I believe this one.
Wait, sorry, maybe I'm not sharing.
You're sharing, but it's just a small screen. But hopefully, yeah, Ben can see it.
Wait.
Yeah, so I think that was the slide you were interested in, Ben. I'm gonna give a quick call out for any other questions for Enrico. And we can hold the meeting on for another couple minutes if, Enrico, you're also free and able to answer any last questions. This is always the point when I say any last questions and no one says anything. I'm glad to have helped chair this session today. And Enrico, thank you so much again for presenting about this work, and we'll be sure to follow up and see how we can all make use of the tools you shared today. Final call out, any more questions? If not, we will wrap up for today.